Just because you're not hearing about something doesn't mean nothing's happening. Well, hello and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam. I've been living with multiple sclerosis for 39 years. And you know that one of the things I have followed with great interest, as I'm sure many of you have as well, is remyelination, which is basically the repair and restoration of the myelin sheath that surrounds each of our nerve fibers, which in MS tends to be much deteriorated as the disease progresses. And I was reminded recently of a clip from a video I watched from Dr. Gavin Giovannoni.
So, the Holy Grail is taking somebody who's got established MS and giving them a treatment to, recover their function. And it's based on the hypothesis that people with MS have demyelinated axons, and you can repair those axons by remyelinating them. I think that's kind of naive because, people with more advanced disease, when they've lost their axons, you can't remyelinate them.
And of course, as you know, I have the greatest respect and trust in what Dr. Giovannoni has to say. And at the same time, I'm reminded of what President Ronald Reagan used to say, trust but verify. So I decided, okay, well, it's been a few years. What's been going on with remyelination? Anything new? I went out and checked, and sure enough, I found some things that I'd like to share with you today. Now, this first one that I want to look at with you is something that obviously is not going to affect our daily life in any time real soon, but I think we sometimes underestimate the importance of these kinds of research projects that understand more why things happen and how things happen, rather than just jumping quickly to some drug that we could do or some treatment we could give that would change the symptom or change the course of something. We kind of want to know why. And a study that's being done right now out of the Oregon Health and Science University, which is where I go for my neurology, is that it sounds like it's going to be really fruitful, and I'm pretty interested in how it's going to shape out. The article here is called Study Reveals Function of Little Understood Synapse in the Brain, and was published on January 12th of 2024, so very recent. And it has to do with these cute little fish, the zebrafish. If you ever had an aquarium or know anyone who has, chances are really good that the aquarium had zebrafish in it. So they're pretty simple little critters. But according to this caption, a new OHSU-led study using zebrafish for the first time reveals the function of a little understood synapse connecting neurons with oligodendrocyte precursor cells in live tissue. In the future, researchers say it may be possible to leverage the synapse in developing new treatments for multiple sclerosis, neurodegenerative conditions such as Alzheimer's disease, and even glioma. And then we read new research from Oregon Health and Science University, which is, by the way, in Portland, Oregon, for the first time reveals the function of a little understood junction between cells in the brain that could have important treatment implications for conditions ranging from multiple sclerosis to Alzheimer's disease to a type of brain cancer known as glioma. The study published today in the journal Nature Neuroscience. And I'll put the link down below for that. We won't go through that paper because I think this article is going to give us the gist of it, but I will certainly link it so that you can read more if you would like. And they say neuroscientists focused on the junction or synapse connecting neurons to a non-neuronal cell, known as oligodendrocyte precursor cells, or OPCs. OPCs can differentiate into oligodendrocytes, which produce a sheath around the nerves known as myelin. Myelin is the protective sheath covering each nerve cell's axon, the thread-like portion of a cell that transmits electrical signals between cells. The study found that these synapses play a pivotal role in producing that myelin. And this is the first time I think that any study I've ever heard of is looking into what part of the body produces myelin. We've just kind of assumed, okay, we got nerves, we got myelin sheaths on them, that's nice. But we also know that the body repairs itself. The body produces myelin. Even those of us with MS, the body is still producing some myelin. How is that happening? And that's what they're looking at here. This is the first investigation of these synapses in live tissue, said senior author Kelly Monk, PhD, professor and co-director of the Volum Institute at OHSU. This gives us an understanding of the basic fundamental properties of how these cells work in normal development. In the future, we might look at how they function differently in the context of MS patients. So what we're looking at here is basic science. And when you're looking at basic science, you can just pretty well assume that it's pretty far from being something that's going to be incorporated in some kind of treatment or pharmaceutical or whatever, because basic science is looking at principles, not products. They say that the fact that these synapses exist at all was the subject of a landmark discovery by OHSU researchers at the Volum, published in the journal Nature in May of 2000. Until that point, synapses in the brain had been known only to carry neurotransmitters between neurons, so the discovery of a synapse between neurons and OPCs came as a revelation. And that's what basic science does. It moves us forward in very fundamental ways. And that's certainly what's been happening here. After 2 decades, we still didn't know what these synapses do, Monk said. Scientists tackled the problem by using single-cell imaging of live tissue in zebrafish, whose transparent bodies enable researchers to see the inner workings of their central nervous system in real time. Using powerful new tools in imaging, pharmacology, and gene editing, researchers were able to use neuron OPC synapses to predict the timing and location of the formation of myelin. The findings are likely the tip of the iceberg in terms of understanding the importance of these synapses, said lead author, I am not going to pronounce that correctly, I'm sorry, but there it is, Jiaxing Li, PhD, a postdoctoral fellow in Monk's lab. Oligodendrocyte precursor cells comprise about 5% of all cells in the brain, meaning the synapses they form with neurons could be relevant to many disease conditions, including formation of cancerous tumors. Lee noted that previous studies have suggested a role for OPCs in a range of neurodegenerative conditions, including demyelinating disorders such as MS, neurodegenerative diseases such as Alzheimer's, and even psychiatric disorders like schizophrenia. So as I say, this is fundamental research because the applications, when they come, are going to affect a number of disciplines, not just one. So by demonstrating the basic function of the synapse between neurons and OPCs, Lee said the study may lead to new methods of regulating OPC function to alter disease progression. For example, these synapses could be the key to promoting remyelination in conditions such as MS, where myelin has been degraded. In MS, this degradation can slow or block electric signals required for people to see, move their muscles, feel sensations, and think. There may be a way to intervene so you can increase the myelin sheath, he said. See, this is exciting stuff to me. Monk said the discovery may be most immediately relevant to cancer. And, that's often the case. I've done several videos recently where something that was originally developed to aid in cancer is being repurposed and looked at for multiple sclerosis. The mechanisms are similar enough that there seems to be more crossover than maybe at first is obvious to us. And they say in glioma, these synapses are hijacked to drive tumor progression, she said. It may be possible to modulate the synaptic input involved in tumor formation while still allowing for normal synaptic signaling. Even though these precursor cells comprise roughly 5% of human brain cells, only a fraction go on to form oligodendrocytes. It's becoming pretty clear that these OPCs have other functions aside from oligodendrocytes, Monk said. from an evolutionary perspective, it doesn't make sense to have so many of these precursor cells in your brain if they're not doing something. Their synaptic connection to neurons, therefore, likely plays a fundamental role in the brain and is worthy of further exploration, she said. In addition to Monk and Lee, co-authors include Tanya Miramontes of OHSU and Tim Chopka, PhD, of the Center for Clinical Brain Sciences in the University of Edinburgh in the UK. Now, maybe fundamental research is not that thrilling to you because you're wanting to just, let's get on with it, let's get something out for everyone in the MS community to actually benefit from. I guess I can't really blame you for that. Well, here's what's going on. According to a newsletter that I found, News Medical Life Sciences, new adaptive clinical trial aims to reverse neurological damage caused by progressive multiple sclerosis. Okay, I'm very excited about that. And this is coming out of Australia. And they say that the first ever adaptive clinical trial for Australians living with MS will seek to reverse neurological damage caused by progressive multiple sclerosis. With an initial $4 million in funding, the trial, commencing in January 2024, will enable researchers to investigate the potential benefits of several medications simultaneously, giving hope to the cohort of people living with one of the most debilitating forms of MS and who currently have extremely limited treatment options. Progressive MS is a clinical form of MS characterized by a progressive worsening of symptoms and disability without periods of recovery. This complex autoimmune and neurodegenerative condition manifests differently in individuals over time, but without treatment, disability steadily accumulates. Working with a national and international group of clinicians and researchers, the multi-arm, multi-stage, and I guess that's the acronym, MAMS, Adaptive Innovative Design, known as Platypus, which stands for Platform Adaptive Trial for Remyelination and Neuroprotection in Multiple Sclerosis. They must have had tons of fun coming up with that, don't you think? Will test two repurposed drugs alongside each other, providing more timely results about whether the treatments are working. MSWA is the leading funder of the trial, having contributed $3 million to Platypus. MSWA CEO Melanie Keeley said, MSWA is proud to fund the ambitious research project, which will transform the way we test treatments for progressive MS. And wouldn't that be neat? And then she goes on to say, Platypus has the potential to deliver a significant breakthrough as we aim to provide real-life outcomes which positively impact the lives of people living with progressive MS, which is always our focus. By testing two repurposed drugs, we hope to find treatment which can be quick to market for the people we support, Ms. Kiley said. That's excellent news. I can almost get excited about that. I want to know more, of course. But so far, it sounds like they're really trying to fast track this and get it out to market so that those of us with progressive MS don't have to wait 20 years, because some of us don't have 20 years. MS Australia CEO Mr. Rohan Greenland said currently, despite a number of traditional clinical trials for progressive MS in Australia, there is no treatment that repairs damage in progressive MS. With an estimated 40% or around 13,000 people living with progressive MS, the launch of Platypus today is a major milestone, the first adaptive clinical trial for MS in Australia. This will ensure a treatment opportunity for people with progressive MS, the greatest unmet need in the MS landscape. Well, hear for that, and I'm really, really glad to know that they're focusing almost exclusively on the progressive forms of MS. It's high time, right? Platypus is an extension of the Octopus Optimal Clinical Trials Platform for Multiple Sclerosis. Okay, I'm definitely thinking they're having fun with these acronyms. Clinical trial, funded by the UK MS Society and launched April of 2023. Neurology professor Simon Broadley from Griffin University's School of Medicine and Dentistry and chief investigator of Platypus says the funding was critical and the new trials will allow the testing of multiple potential therapies simultaneously and bring results much faster than traditional clinical trial methods. Collaborating with our octopus partners in the UK, we'll be trialing the drugs metformin, which is typically used to treat type 2 diabetes, and alpha-lipoic acid, which is a health food supplement. These therapies have shown promise in promoting neuroprotection and/or myelin repair in MS, Professor Broadley said. And just as an aside, I know about using metformin. They're looking into whether it might have some benefits for people with MS. I have heard about that. I don't know a lot more about it. So this is interesting. And then alpha-lipoic acid, I've been taking that for quite a long time based on a study that, by the way, was done out of Oregon Health and Science University that showed in clinical trials that people seem to benefit from alpha-lipoic acid. So I've been taking that for, 5-6 years now. The platypus trial will be rolled out across Australia through a collaboration of 20 academic and healthcare institutions and aims to recruit more than 250 participants in Australia. That's a good-sized clinical trial. I really appreciate this. They're doing that. By the way, if you live in Australia, you might check this out, because if they're in active recruiting right now, if you've got progressive MS, you might very well be eligible for this trial. These multi-arm, multi-stage trials will require less time and fewer participants to assess the efficacy and cost-effectiveness of metformin and alpha-lipoic acid, Professor Broadley said. The platypus team includes leading internationally recognized neurologists, neuroscientists, statisticians, and health economists covering 5 states across Australia, as well as people with progressive MS. Nigel Caswell, diagnosed with MS in 1993 and MS Australia's John Study Award winner in 2022, is excited for those people living with progressive MS in Australia. The launch of Platypus today provides extraordinary hope for many people living with MS and is a major moment for MS research, Mr. Caswell said. President of MS Australia, Associate Professor Des Graham, says he is personally and professionally delighted for Australia's MS community that Platypus can now be realized. With a $3 million grant from MSWA and a $1 million grant from MS Australia, this revolutionary adaptive trial provides tremendous promise for those living with progressive MS in Australia, and MS Australia is extremely proud to lead this initiative, Associate Professor Graham said. MS Australia's head of research, Dr. Julia Morahan, says Platypus will investigate whether these drugs reduce brain shrinkage and clinical progression of MS. And that's actually what alpha-lipoic acid was purported to do, was to slow brain shrinkage. So that kind of explains the connection, at least with one of the 2 drugs that they're looking at. We hope that this will lead to positive trial outcomes that are readily translatable into practice, providing new hope for improved care for people with progressive MS in Australia and beyond, Dr. Morahan said. And the source of the article is out of MS Australia. Well, in addition to the articles, here's something that Dr. Ben Thrower says is coming down the pike. And I think we've heard something about this before. And he seems to think that it's going to be quite important in the treatment of MS.
I just read about a study published by Pfizer in 2021 in which they'd reported using an mRNA vaccine to stop MS in mouse models. So wanted to know what you know of how that modified mRNA therapy route is progressing.
So it's still in testing. So that that was one of the positives of the whole COVID experience, is we did kind of spin off this messenger RNA technology. So what they're doing with the MS vaccine with messenger RNA is they're using messenger RNA that codes for a myelin protein. So they're having cells in the human body produce a little more myelin protein in the hopes that it actually teaches your immune system to tolerate myelin protein. Sort of like, getting an allergy shot. So you're desensitizing yourself, say, to dog hair or ragweed. So you're basically desensitizing your immune system to myelin. So works in the animal model. We always have to be cautious. There are a lot of things that have worked in the mouse that didn't translate into humans, because ultimately the mouse's immune system is not a human immune system. But fingers crossed that we see something positive out of this.
And then one more thing I'd like to say. There is a clinical trial ongoing at Oregon Health Science University, conveniently located only a couple hours away from me, and it's one that I might even be interested in being involved in. So I have sent a message off to the coordinator of that study to find out if I can be involved. It has to do with aerobic activity and remyelination. And it looks pretty simple. Let's go over some of the details just really briefly. If you want to have all the detailed information, you can go out to look at the clinical trial itself. But I'm just going to cover a couple of things. Basically, there are two groups that are going to be involved in this clinical trial. One arm is going to be the experimental arm, and they call that the MS cycle arm, and that is 22 participants in the clinical trial arm will be randomized to MS cycle, an aerobic exercise intervention on a stationary ergometer. Participants will exercise thrice weekly for 30 minutes with graded supervision for 24 weeks. So it's a pretty lengthy program. But the control group will be the active comparator, and that'll be 22 participants in the clinical trial, randomized to MS, take control, a monthly hour-long MS education control group led by a trained facilitator. So what I kind of like about this one is it doesn't matter whether you're in the control or if you're in the actual experimental arm, because you're going to get some benefit. Some clinical trials, the control group is basically a placebo. But obviously with exercise, you can't do a placebo that's very convincing. People will know if they're exercising or not. This looks like one that could be beneficial whichever arm you're in. And then down below there are the contacts. And I have already, as I said, sent an email in. And in fact, they have responded to me already. So that tells me that they're actively recruiting and interested in gaining more people to participate. I will let you know if anything comes of this. Since I live 2 hours away, it may end up not being practical for me, but I'm curious enough. I want to see. And then just so you can see, the main location and the only location is Portland, Oregon, at the Oregon Health Science University. But also you might be interested in knowing who the sponsors and collaborators are. And we have here Oregon Health and Science University, Eunice Kennedy Shriver National Institute of Child Health and Human Development, the Portland VA Medical Center, so Veterans Affairs, Oregon Critical and Translational Research Institute, the Myelin Repair Foundation, the EMD Serono Company, which is the pharmaceutical, and then finally Medical Research Foundation of Oregon. So kind of a variety and quite a number of collaborators and partners, and that also makes for a good study in my view. Okay, well, I think what I'm trying to say here is that despite my agreement with Dr. Giovannoni that it would be naive to think that remyelination technology is going to be available to us anytime soon, it doesn't mean there's no hope, and it doesn't mean that there's absolutely nothing that we can do in the meantime while we wait. So in the interest of ending things on a positive note, here's what Dr. Stephen Hauser has to say. And Dr. Hauser, as you know, he's written a book recently. He is a pioneer in the development of disease-modifying therapies for multiple sclerosis. And it's always a pleasure to know that someone who knows that much about what's happening in the field has an overall positive outlook.
By understanding now the cells and the genes that are driving progressive MS, I think we will have better therapies for people whose MS has been with them for decades and who have significant progressive worsening. I think that there's an opportunity to develop, for the first time, truly effective repair therapies. Therapies that can save injured nerve cells or regrow damaged myelin, and that could truly make a difference for people with disability today. There are also great opportunities to use stem cell biology and artificial intelligence and machine learning and computer-brain interfaces to find additional ways to help restore function in people who have lost function. So I understand that for people with MS that has caused significant disability, prospects sometimes seem bleak. But I think that answers are on the way.
Well, as I said, on that note, we'll end things. And that's all I have for you today. But I would love your comments about this. Do you think remyelination will be a reality in time to help you with your MS? Let me know in the comments below. And I'll see you again in my next video. But in the meantime, of course, take really good care of yourself.